And I'll be presenting a high-resolution deconvolution of RNA secondary structures via long-read nanopore technology. And I like to start every talk with a very bad joke. These are examples of 4 different white bears that are genetically distinct, none of which are albino, but only one of them gives talks. So I work with nanopore sequencing, and I use it to look at RNA secondary structure. And what we do in our experiments is try to approximate what we see with shape map, but in a more accessible manner and with more detailed information. So we apply a structure probe called the acetaminophenol, which binds to the 2' hydroxyl on the RNA backbone and has like no specific base preference to our sample RNA. And then we sequence them with nanopore base callings, and we detect where we see modifications in the nanopore signal. So this is a basic overview of the experiment. It gets way more complicated, but I think this is a good way to think about it. And just to prove that I'm not pretending to do this, we have a software pipeline called currently Dashing Turtle. And we looked at our control samples, including the HCV IRAs with shape map and also with nanopore Dashing Turtle. And we compared how well these annotations were detected with shape CE, shape map, DT, and just the overall secondary structure that we observed. And not just in this example, but in many examples, DT with nanopore was comparable to the shape map experiments. Additionally, we actually saw improvement in some of the long single-stranded regions and with bulge detection, and we saw this across our control samples. Right now, we're also investigating how well our probe looks at kink turns in the secondary structure, just because we have a very nice small probe that can easily access different parts of the structure. And to test this out in more detail, we looked at something called natural antisense transcripts, which are thought to bind fully complementary in the 3' UTR regions. The thing is, we do not know exactly how these bind, and so we were kind of investigating these with nanopore, and it's such a short region that more difficult experiments wouldn't be able to resolve it fully. So we looked at 2 of these called SEN and IK2. And one of the first steps in the overall method of looking at the interaction landscape is to look at the reads that we get from nanopore. So here we have about 2,000 reads sequenced for each of the molecules in the interaction space, where you have the darker areas are representing higher reactivity, and you can see it varies across the landscape. So it's not always deterministic, and it's not always going to have one conformation in a sample. And we see that for both of the molecules, which means there's different binding interactions and conformations happening. So to investigate that, we first looked at the clustering using the Hamming distance of the predicted reactivities from Dashing Turtle, and we formed these clusters. Then we used the distance matrix of the clusters to see how similar or dissimilar the clusters were. And we want to do this to select the optimal representations that are occurring in the RNA-RNA interaction landscape. So we looked at this as well as, I think I clicked it too often, or not enough. Sorry, it's a bit slow. I think it's just loading. Oh, okay. I'll skip the other one that took forever. So we also looked at the correlation coefficients between those predicted interactions, and we select optimal clusters that represent what's happening in the RNA-RNA interaction landscape. So we go from the interaction landscape to the clusters to the centroids, which are representatives of the optimal RNA configuration for a given cluster. And we choose K of these, which could be any number that you think is best representing the landscape. And we use these centroids or representative molecules to model the interactions in phase and or time. So in any interaction that you have, there's a sample space, and it's not just reserved to the 2 molecules that you're interested in. So here we have A and B, and of course, we're going to assume that A and B are interacting, but you're going to have intramolecular interactions with A interacting with itself, and you're going to have hybridization with copies of the same molecule. So you'll see that throughout the landscape. And we're not going to be able to observe all of these interactions. We can really only clearly see a few interactions at a time. So we really have to think about ways to model the space so that we can represent these interactions better. And to do that, we looked at the probability of interacting for each of the different types of molecules in the sample. So for each nucleotide, for each representative molecule, we calculate a probability of it binding to itself, to an interacting molecule, or like an intramolecular binding. And for each of these, we do it for all the molecules in the sample, and we compare it to the clustering representative molecules in the target sample. Now, as you can imagine, this gets very large, very fast computationally, and becomes a very complex space. So we like to represent our beliefs about some of the bindings that we can't observe directly using deep neural networks to approximate the uncertainty in the states and the history of the state space. So overall, this is a variation of reinforcement learning applied, but it's very specific using a partially observable Markov decision process. Basically, this is talking about the states that we can't see and how we represent the steps or the phases that we would need to touch upon to get to our final interaction state. And so a partially observable Markov decision process, or POMDP, allows us to investigate those transitions. And so we have preliminary results. So I guess the first preliminary results that we looked at were the conservation of the reactivity of the molecules in the sample. So we noticed that SEN was very highly reactive and very single-stranded for most of the time in the interaction landscape, whereas IK2 actually preferred to conserve a lot of its base pairings. And so when you combine that, you can kind of see phases of how this molecule is actually interacting in the space. So here we can see that SEN almost completely unwinds to accommodate IK2 in one of its dominant phases. So this is one of the dominant clusters in the landscape, whereas IK2 is kind of maintaining its structural presence. And then we can see the reforming of these additional interaction pairings to get to like a final state. So we know natural antisense transcripts are expected to bind in a fully complementary way. And so we can get to another intermediate state or another intermediate phase in the sample that looks more like this, where we see these holes in the fully complementary region that they could be kink turns that were annotated with the chemical probe, or they could be incomplete bindings. So we can see a vacillation between this state and the final expected state in the sample. So we can go back and forth a little bit in those structures. And what our method allows us to do is kind of view those intermediary states. Now, we didn't do time-based experiments in this, but if you wanted to do this on a time-based level, you could actually time your nanopore sequencing for different points in an interaction and actually look at different phases of how the structures might interact. Additionally, I'm not displaying it here, but we have the other interactions as well in this space. So we can look at what's happening when these molecules are not just binding with each other, but they actually do have other bindings that we've detected for other parts and other regions of the RNA. So overall, this is kind of what the pipeline looks like right now. And here's a cartoon turtle. Dashing turtle. So dashing turtle can also be weighted by stoichiometry. So we can say that, for instance, in our samples, there are more SEN molecules present, and so certain interactions are more likely to happen with SEN in certain instances. We can also model and hypothesize about interactions that cannot be directly observed, and that's where the POMDP comes into play. And I think that's a very nice feature, because I don't see it very often in the interactions that I've seen that they bind exclusively with each other. So in protein language, I guess there would be like more promiscuous domains. And we can also show transitions over time or phase of complex formation. And so that's basically what I want to say, but I wanted to acknowledge my direct collaborators at IRCM, Gregor de Bishop, Juan Carlos Padilla, and Eric Loquier. They did most of the experimental work and have been very helpful. My supervisor, Jérôme Modespil, who you've seen earlier, the Waldenström Group, my lab, which have been really awesome, and esteemed collaborators, and Panti and Vladimir Reinhardt. I can be reached at this email address if you have additional questions. Any questions?
Could you also model RNA binding proteins binding onto the different intermediates?
Yeah, you actually can. Oh, sorry. Yeah, you can actually model the RNA binding proteins as well. You can see when you actually the structure probe to the bound molecules where bindings are not occurring and how often that occurs. So you could actually even calculate a frequency of binding for the binding proteins. And I think that's a big area of interest right now with nanopore, is looking at protein binding and then the RBPs as well.
Okay, quick.
So for looking at different conformations of RNA structure, what can you do with this particular tool, or I guess in general?
So we know that RNA interactions are not happening in a deterministic fashion every single time, right? So in a sample, you have like a mixture and it's heterogeneous, and the bindings are very diverse. So sometimes it's going to be intramolecular, sometimes it's going to be intermolecular, and sometimes it's it's going to hybridize with the other molecule in the sample. But we don't know that. And over the course of the entire RNA strand, it can differ substantially, right? You can have one part that's binding intramolecularly, and you can have another part that's binding with a copy of the same molecule. So this tool actually lets you see that and what we can hypothesize different manner, as well as looking at kind of the experimental data to validate that. So it's not just like an RNA fold calculation. We actually combine that with the experimental reactivity to get really nice probabilistic representations of those interactions. So it's a much bigger and I would say more diverse landscape of an RNA-RNA interaction.